Hello, and welcome to this video lesson on the National 5 biology topic, Absorption of Materials. We are still on Unit 2, Multicellular Organisms, and this is Topic 7, Absorption of Materials. Once again, here's the SQA course specification or mandatory knowledge section for this topic. Remember, this is all of the bits of knowledge and content that you could be tested on in a test or exam at this level for this topic. I'm going to break down this topic into smaller sections so that we can understand it properly. As we go through the theory for each section, there will also be some questions after each part so that you can be sure that you're ready to move on. So our learning intention for this part is to learn about absorption of materials in the body. And hopefully by the end of this lesson, you'll be able to explain why exchange systems are required in animals, explain the need for capillary networks in tissues, describe the 3 common features of surfaces involved in absorption of materials, and describe the features of the alveoli in the lungs and the villi in the small intestine, which make them efficient at exchanging materials. So first of all, we're going to look at absorption in general. Now first, let's ask ourselves the question, why is absorption important in the body? Well, when we spoke about respiration in Unit 1, KA7, we discussed the fact that to do aerobic respiration, cells must have both oxygen and glucose. In our last topic, we also discussed how the blood carries oxygen and nutrients to cells within tissues that require them. So oxygen and nutrients from food must be absorbed into the bloodstream. The reason this absorption is important is because both oxygen and nutrients must be delivered to cells in order for respiration to take place. Additionally, this isn't just about cells gaining materials such as oxygen or nutrients, they also need to get rid of waste materials such as carbon dioxide. Carbon dioxide and other waste materials must be removed from cells into the bloodstream. Now that we know what must be absorbed into the bloodstream, we must think about where in the body absorption happens. In the cases of the 2 gases I mentioned, oxygen and carbon dioxide, they are exchanged in the lungs. Within the lungs, oxygen is absorbed into the bloodstream and carbon dioxide is released from the bloodstream and into the lungs. This exchange is why we breathe in oxygen into our lungs and breathe out carbon dioxide. Nutrients, on the other hand, are absorbed into the bloodstream within the small intestine. These nutrients come from the food that we eat and are broken down within the digestive system, and the resulting nutrients are absorbed within the small intestine into the blood. We're going to discuss both of these in more detail later in the lesson. In our last topics, we spoke about the 3 different types of blood vessels: arteries, veins, and capillaries. Since absorption relies on diffusion, blood capillaries are essential for the exchange of materials. If you remember back to KA6, tissues that are made up of cells contain capillary networks to allow efficient exchange of materials, so absorption cannot happen without capillaries. So let's try some quick questions on what we've covered so far to check our understanding of this topic so far before we move on. So pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. Okay, so the first question asked, nutrients from food are absorbed into the, and the answer is bloodstream. Number 2 asked, in which organ of the body does this happen? The small intestine. And number 3 asked, within the tissues of the lungs and the above organ, which vessel allows the exchange of materials? And that is the capillaries. Okay, so surfaces involved in the absorption of materials have 3 features in common with one another. They all have a large surface area, they all have thin walls, and they all have an extensive blood supply. These all increase the efficiency of absorption. Now this slide explains each of these common features in a little bit more detail, but this is just extra just to help you understand why they increase the efficiency of absorption. The larger surface area for absorption, the more materials can pass by diffusion into those cells. Thin walls allow very quick diffusion of materials into the blood or tissues. And finally, all absorption surfaces have an extensive blood supply, which allows more opportunities for body cells to contact a capillary network. The first thing we're going to focus on in the body, which is involved in absorption of materials, is the lungs. The lungs are gas exchange organs, meaning the lungs and the blood swap oxygen and carbon dioxide. Lungs consist of a very large number of alveoli. You can see the alveoli here. This is the lungs on this side on the left, and if we zoom in, this is a bunch of alveoli, and you can see the dark blood capillaries over the top of each, giving them a good blood supply. If we zoom in again, you can see a single alveolus, so this part here, with a blood supply going around the outside, so a blood capillary moving around the outside. Your lungs contain about 600 million alveoli inside of them, and by having this large number of alveoli, they provide a very large surface area for gas exchange. Remember, a large surface area was our first common feature of surfaces involved in absorption. Here's a diagram showing the role of the alveoli within the lungs in gas exchange. You can see the bundles of alveoli that are highlighted here, and the blood capillaries going around the outside in close contact with them. You can see that the blood comes into the lungs is deoxygenated on this side, and then the blood that leaves the lungs is oxygenated. Essentially, we breathe in oxygen, like you can see here, into the lungs. This oxygen then enters the alveoli at the bottom here and passes through the alveolar walls into the blood capillary, and then the blood leaves the lungs oxygenated. Carbon dioxide has the opposite journey. Blood that enters the lungs over here will have a high concentration of carbon dioxide as it's picked up this waste product from cells within the tissues. As the blood capillaries move over the alveoli, the carbon dioxide diffuses from the blood capillary into the alveoli, and then the carbon dioxide leaves the lungs as we breathe out. So oxygen and carbon dioxide are both absorbed in different directions through the thin alveolar walls. The alveolar walls must be thin or diffusion wouldn't occur. Remember, thin walls is the second common feature of surfaces involved in absorption. Finally, there are many blood capillaries associated with the alveoli, which means they have an extensive blood supply. This is the third common feature of surfaces involved in absorption. This table at the bottom summarises the direction of movement of oxygen and carbon dioxide in the lungs, which I explained on the previous slide. So oxygen moves from a high concentration in the alveoli to a low concentration in the blood capillaries, and carbon dioxide moves from a high concentration in the blood capillaries to a low concentration in the alveoli. Now, it's very important to understand the direction of movement of carbon dioxide and oxygen in the lungs, as a lot of questions rely on you being able to apply this knowledge to these types of diagrams. Here you can see a single alveoli with a blood capillary going around the outside. You can see that deoxygenated blood coming in and oxygenated blood leaving. Remember, the blood that comes in has a low concentration of oxygen and a high concentration of carbon dioxide. The carbon dioxide moves from a high concentration inside the blood capillary to a low concentration inside the alveoli by diffusion and is then released as we breathe. The oxygen moves from a high concentration in the alveoli to a low concentration in the blood by diffusion, and therefore when the blood within this capillary goes to leave the lungs, it will have a high concentration of oxygen and a low concentration of carbon dioxide. Here is an example of a past paper question using this type of diagram. In a question like this, 4 areas have been labelled. The alveoli itself is A, blood as it enters the lungs is B, blood where it's passage through part of the blood capillary around the alveoli is C, and D is when the blood is ready to leave the lungs. This question would ask about where a certain concentration of oxygen or carbon dioxide would be found. For example, it may ask at which position is the concentration of oxygen in the blood highest. Now, one thing to notice there is that I said blood, so it cannot be A, as that is the alveoli, so it has to be either B, C, or D. The highest concentration of oxygen in the blood would be as it was leaving the lungs, so that means it must be D is the correct answer. If I asked instead at which position is the concentration of carbon dioxide in the blood the highest, again I can get rid of A as that's not the blood. Blood that is arriving at the lungs would have the highest concentration of carbon dioxide, which is a waste product, so the answer would be B. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. So question number 1 was, why do tissues contain capillary networks? The answer is to allow exchange of materials at a cellular level. 2 was to name the 3 things that surfaces involved in absorption materials have in common. They are a large surface area, thin walls, and extensive blood supply. 3 was give 3 features of an alveolus, which makes it efficient at its role of gas exchange. Again, it's the same thing. So large surface area, thin walls, and extensive blood supply. If you're ever unsure on an answer for a question for this topic, just try and go for one of those and you'll usually be right. Okay. So So the second and final area that we're going to focus on in the body, which is involved in the absorption of materials, is the small intestine, where we find the villi. Light gases are absorbed in the lungs. Nutrients from food are instead absorbed in the small intestine. They are absorbed within the small intestine into structures called villi. Here you can see where the small intestine is found in the human body, which is over here. So this is part here, right in the middle of the large intestine that's going around the outside. The small intestine is in the middle part here. If I took a cross section of the small intestine, so cut it across the way, this is what it would look like on the left. It's not just a simple tube, it contains millions of these tiny structures called villi. Every square inch of your intestine contains between 6,000 and 25,000 villi, and they're extremely important for the small intestine's function, which is absorbing nutrients. So here's a diagram of the small intestine on the left, and if we zoom in on one part, you can see the villi here, and if we zoom further, you can see this single villus, which is what we're going to be talking about in more detail over the rest of the lesson. Now, just like the alveoli in the lungs, the villi in the small intestine share the three common features found in surfaces that are involved in the absorption of materials. Firstly, there are an extremely large number of villi within the small intestine, which provides a large surface area. They're all thin-walled, and each villus contains a network of blood capillaries, which gives it an extensive blood supply. However, villi also contain a lacteal as well, and both the blood capillaries and the lacteal are used to absorb the products of digestion. Here you can see the structure of the villi in more detail. You can see the thin wall that goes around the outside of the villi in order to aid diffusion. You can see the lacteal, which is this part in the middle, and then you can see the blood capillary running between the lacteal and the thin wall. So before we go on to look at the specific nutrients which are absorbed by the lacteal and blood capillaries, we must first revise the products of digestion. The digestive system breaks down carbohydrates, proteins and fats into smaller products which can then be absorbed by the villi within the small intestine. Carbohydrates, proteins and fat, as they are too large to get absorbed by diffusion, so they have to be broken down into smaller products. This topic links with Unit 1, KA7, Proteins and Enzymes, where you learned various enzyme degradation equations. You should remember that each equation starts with a substrate, has an enzyme above the arrow, and the products go at the end. In the case of carbohydrates, we normally ingest starch. Starch is broken down by the enzyme amylase in our body into maltose. Now, you must say maltose for Unit 1 KA4 questions. However, for the purpose of this topic, we're going to talk about the product of carbohydrate digestion as being glucose, which is a product of maltose digestion. So maltose is actually still too big to be absorbed, so it has to get broken down further into glucose. In the case of proteins, they're usually broken down by the enzyme pepsin to create smaller polypeptides, which can then be broken down further into amino acids. Finally, at the bottom, fats are broken down by the enzyme lipase into fatty acids and glycerol. So now we know the three products of digestion. Glucose is the product of carbohydrate digestion, amino acids are the product of protein digestion, and fatty acids and glycerol are the products of fat digestion. Now we need to learn where each of these products is absorbed within the villi. So here's a common diagram of the villi in the small intestine. In dark grey in the middle, you can see the lacteal, and in white around the outside of it, you can see the blood capillaries. Now we need to learn which of these absorbs each of the products of carbohydrate, protein, and fat digestion. So, first of all, if we look at the lacteal absorbs the products of fat digestion, so fatty acids and glycerol, whilst the blood capillaries absorb both the products of carbohydrate and protein digestion, glucose and amino acids. You must remember which of these products are absorbed by the lacteal and the blood capillaries, as this is a really common question. Another common question is, where do you find in this diagram the highest concentration of, and then an example of one of these. So here I've added 4 multiple choice options, A, B, C and D at the bottom of the screen. You can see here that the blood in the blood capillaries and the fluid in the lacteal move towards the right of my screen. So if I asked where the highest concentration of glucose was found, well, I know that glucose is found in blood capillaries and not the lacteal. So I can straight away eliminate C and D because they are blood capillaries. A is where the blood enters the villi and small intestine, so it won't have picked up much glucose yet. So the highest concentration of glucose must be after it's been absorbed through the thin wall here and as it's leaving. So the answer is B. Now, if I asked instead, where is the low concentration of glycerol? Well, I know that glycerol is a product of fat digestion, so it's absorbed in the lacteal, so I can get rid of the two options that are blood capillaries, so A and B. Out of C and D, the fluid enters the lacteal on the left with low fatty acid and glycerol concentration and leaves after absorbing them, which will now have a high concentration. So a low glycerol concentration would be found at C. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions either by saying them out loud or writing them down. And when you're ready, play the video and we'll go through the answers. First question was, in villi in the small intestine, where are the products of carbohydrate and protein digestion absorbed? They're absorbed in the blood capillaries. In villi in the small intestine, where are the products of fat digestion absorbed? They're absorbed in the lacteal. Name the above products. So if I was to name the products of carbohydrate digestion, that's glucose. If I was to name the product of protein digestion, that's amino acids. If I was to name the product of fat digestion, that would be fatty acids and glycerol. And my final question was, give 3 features of a villus which makes it efficient at its role of absorbing the products of digestion. Well, they are the 3 common features that we talked about for the alveoli. So thin walls, large surface area, and extensive blood supply. So that's us finished learning about absorption of materials. I hope that you're now confident about why exchange systems are required in animals and the importance of capillary networks, and that you can now describe the key common features of surfaces involved in absorption of materials using the alveoli and the villi as examples. This is quite a difficult topic, so please feel free to go back and watch parts of the video again in the future if you need a refresher on any of it. Thank you once again for listening.